Okay, so here we go again. Let me get your frame of reference for you. Am I moving? No, I'm not moving the pointer anymore. Here's D1. Yeah, and then shift over to the next space. That's D2. Shift over to the next space where 1350 is. That's D3, and then D4. Now it's not showing, but the segments are these vertical lines. So segment 1 through 6 are here in each one of these domains. And I've mapped the mutations on there. And what I want to tell you now is we've done this unstructured analysis. We do a scan across the entire sequence, and we ask if there's a statistical signal of the variant being in a region that is pathogenic in purple, benign in green, complex in blue, or lethal in red. Now let me explain what that means. The pathogenic purple lines are places where the patient population had variants in a stretch across the gene that were not present in the healthy folks, okay? The green ones, the stretch of sequence there represents variants that were present in the healthy population, but not the epilepsy population. The blue lines represent more complex regions where every amino acid could shift to being pathogenic or benign, pathogenic or benign. So it's a complex region. And finally, the red lines are the ones that are really surprised. Those asterisks represent statistical significance. That was a significantly long stretch of sequence where nobody had a variant, neither healthy nor epileptic. So that, to me, suggested that these regions cannot tolerate mutation at all. And I call them embryonic lethals. In other words, if a conception occurred with a variant in these regions, it wouldn't make it for very long in gestation. So that's a hypothesis. It has to be tested, but that seems to be the effect. Now, when we compared SCN8A with 1A and 2A, SCN8A had more of these lethal regions, suggesting that SCN8A is a more important gene in some ways and can't tolerate mutations in certain areas more so than the other genes, which correlates to the slide that Gabi showed you, that SCN8A phenotypes in terms of seizure frequency is worse than in many other epilepsies. So we have our challenges in defeating this disease. It's a bad one, but I think with what we're learning, we have some hope that we're going to get some breakthroughs in the near future. Let me just point out one thing. I said that we might doubt that a variant in this linker was causing epilepsy, but notice they're all falling within this little complex region of blue. So you might ask your doctor or a researcher, they might say, oh, that variant doesn't cause epilepsy, especially if it's inherited. But I would say we need to reconsider that if it's in this region. Here's one that maybe is not. Well, I'm not saying that. This one might be on the edge. But notice that many of the other variants fall into regions where it's purple. Here, I'm over at the front of the gene, 267, 386, 403. They're all landing on purple spots, 850, 845. But then going back to the front, you can see some of them are landing in complex spots, 132, 205, et cetera, as you go down. Over on the opposite end, you know, they got 1872. You might say, well, there's a benign chunk, but look at that complex stretch. All of these variants fall within that complex stretch. So it really depends which amino acid you have that tells us whether it's likely causing the epilepsy problems. So that is what I'm calling the starting point for a more sophisticated analysis comparing people with these variants in these regions with their various symptomology to look for correlations. That's where we're going in the future so that we can really make predictions about what these variants mean for your child and for the doctor who wants to treat your child. Some of these targets maybe some of these stretches may be targetable with unique therapeutics.